So I'm an analytical chemist by training, and I've been working at Genentech for the past 5 years. So I'm in the technical development department, and what I like to say, so we take the nice ideas from research, so most of them that we saw today during the presentations, and then we go through the key considerations that we need to take into account to transform them into real products. So today I'm going to give like an overview just by choosing one molecule and going through some of the key considerations with examples of the thought process that we go through in development. So as we heard today from Oscar and Fadi, so there is a need for speed, so we really need to be faster to be competitive. Having like a 10 years turnaround time to bring medicine to the patient is really, really too long. At the same time, we have increasingly complex formats. So I come from the large molecule department, but I really work more and more with the small molecule department at Genentech, and I think it's the same across other companies. So we have very fancy formats coming down the pipeline, these protein polymer conjugates. So today I'm going to talk about this. We have these peptides that are done in small molecule, but they're proteins, so they need our support. So there is really a synergy that is more and more evident between large and small molecules. So this is also one of the messages that I want to convey here today. And because of this, so the need for speed and large molecule, small molecule, funky formats, the standard technical development practices and also the standard analytical tools and the standard thought processes, they are not applicable as straight as before. So we need to put a little bit more effort in how we're going to present our work to the health authorities. Where's Johnny? There. Okay, so the molecule that I chose today is a multi-arm protein polymer conjugate, and the reason why I can talk a lot about this molecule today is because it's not in our pipeline anymore, so because you're here. Okay, so this is a very interesting molecule. So it's a PEG core. So the PEG was done by the small molecule department, a PEG with 8 arms. And at the end of each arm, there is a protein. In this case, it's a Fab. So the molecule is pretty big. It's about 500 kilodalton. And what are we going to see today is the impact of the different parts that we have to go through during the development. So the first one is the raw material impact. So in order to make this molecule, we have to assemble different pieces together. So we know that the protein is what is going to do the job, but to make this protein, there are like different pieces that need to come together. So I want to show you by choosing one of these parts, what is the impact on the development of the molecule. Then the other thing is some formulation challenges, right? So we know that the molecule is working, but we know that we want to have a specific concentration, and in order to have that concentration, we need to make some changes to the formulation. And then finally, it's the control, right? So when we file an IND for early stage programs, safety is really what we have in mind. We want to give something safe to the patient, and we want to ensure that we have consistent quality when we make our batches, right? So we need to have in place the standard QC testing, the standard controls, and so I want to show you how the standard QC controls need to be adapted when we have unique format molecule. Okay, a little bit of background on this molecule just to help understand a little bit more what I'm going to talk about. So this molecule was thought to treat like an eye disease, is macular degeneration, and the reason why we wanted to make this multi-arm format is because we really wanted to decrease the frequency of injection in the eye. Normally patients with this disease, they have to go for monthly injections, but there's a lot of efforts to be able to have something that can stay in the eye much longer. Well, 4 to 6 months ideally, so the patients only need to go once every 6 months to have the injection in the eye. So that's the background on this format. And then there were some data that we have generated showing that there is really a correlation between the size of the molecule and the time that stays in the eye, so the vitreal half-life, right? So there is like a very clear correlation. This is really what brought to bring this molecule in our pipeline. This is an overview of the process, but what we need to retain here is that we need to bring pieces together, right? So we have our fab, and then through a process, this fab is attached to this peg. So the peg has 8 arms, and at the end of the arm, there's a maleamide. So this maleamide attaches to the fab. So through a conjugation process, then we purify, and then we have our final product. So to make it, it's not so hard, but then one key question that we need to take into account, so what is the quality of the peg, how this is impacting the quality of the final material, and also the safety, right? So in order to make the peg, this is a synthetic process, so solvents are used and reagents. So we need to make sure that whatever is used to make the peg, it's safe. And then if it's not safe, that is cleared away, right? So these are considerations that we need to take into account. So for this specific peg, we want to make sure, do we have consistently the same number of arms? Also, do we have all the arms that have the same amount of maleamide? And then, as I said before, do the peg impurities, they carry through the final conjugate? So this is an example for the peg, but then similar thought process should be used every time you use raw material and then intermediate for your final product. So this is an example. So this is the peg. So when this molecule was done in research, I believe they just ordered the peg from the first vendor that they found online. And then it was great, but then that peg vendor was not able to perform GMP material, so that would not work for us. So we had to go around and see who else out there was able to make comparable quality and also able to make GMP quality material, right? So that took a few months, right? So this is also something that you want to take into account. So the research department really needs to work with the development when there is something new that comes, into the portfolio. Anyway, so we found 2 CMOs, CMO A and CMO B. And then the polydispersity index is something that is important for the PEG. So the first one had 1.33 and the second one had 1.04. This is just a number, but this number makes a huge difference. So we can see in the first one, so if we use MALDI, is a mass spec technique, but is to show that we have different subfamilies in this PEG, and the second one is much more pure. And then when we make our final conjugate, so this is another analytical techniques that we use for size. So you see the first one is way more polydispersed compared to the second one. So definitely we want to have the second one. So the choice was pretty obvious here to go with the second CMO, but this is really put some delay on our timeline, right? So just to make sure that this is really important to make sure you choose well the vendor as early as possible. And then something else, so once we have chosen our CMO, then we needed to make sure that the lots were the same consistently. So if you see here, these are 2 lots, and then there are some differences, and then if you see those differences, you can say, oh, that's not such a big deal, maybe 1% difference. But then actually when we use this material to make our conjugate, this is a size exclusion chromatography profile, so it's size. So you see our major component here, that doesn't really work. And then on the right, in the low molecular weight region, you see that's a peak that's slightly, quite much higher compared to the blue trace. And then this can be an issue. And the reason why this can be an issue is because when you set your specification to release your batches, you put like a specific acceptance criteria for that region. So it's say, I cannot accept more than 4% because I know this can be toxic. And so when you see these differences, you really have a very, very high risk of rejecting your batches, right? So these are all thought considerations, very small differences that can make a huge impact, right? You can make your material and then you have to reject the batches because you have your predefined acceptance criteria that are not met, right? So this is something very, very important that you need to take into consideration. Oops. Right, so that was just one quick, fast example of the impact of the raw material. The other example that I wanted to mention here today is for formulation, right? So as I mentioned at the beginning, this product was going to be for slow release into the eye. So you need to have a high concentration, right? Because if you inject only once every 6 months, you need to make sure you have enough material that's in the eye. So the concentration that our formulation group was targeting was 200 milligram per milliliter. So this is our concentration range. But in that concentration range, the viscosity was extremely high. And then our manufacturability limit is much lower, as you see there. So it's 20 center points. So what happens if we're much higher than our manufacturability limit is that we cannot make it. So we have certain filters when you do the purification, and then those filters only accept a certain amount, right, of viscosity. So if we have something higher, we cannot push it through. Or also, when we have the syringes, so this is going to be injected into the eye, we can literally not push it through the eye, right? So we know that the product is working really, really well. We know we have our protein polymer conjugate, but then we cannot inject it or we cannot make it, right? So these can also cause like some delays. So this is definitely something that requires, some thinking. So in this case, what we did, we wanted to see whether we could find an excipient that could lower the viscosity, or the plan B was to find other types of formulations. So very quickly, I'm going to show you. So we tried D-arginine, and then there were like different concentration of D-arginine, and then you can see that the viscosity can really be lowered. But unfortunately, the D-arginine was toxic at these levels for the eye, so that was really could not be considered. And so what we had to do, we had to use lyophilized formulation. So the liquid formulation, which is the intermediate step, so our drug substance was at a lower concentration, so could be done from the manufacturing standpoint. And then it was lyophilized, and then before giving this to the patient, we would reconstitute with a lower volume, and so the concentration is 200 milligram per milliliters. But all these requires really assessing and studying of all the different steps. So just to show you how the concentration can really impact the development timeline of a molecule like this. Right. The last example is the QC testing. So in the department where I work, we see molecules from early development to post-approval. And so definitely the criteria that we apply for early stage development QC release are less stringent compared to post-approval and late stage commercial. But we do have QC testing, GMP testing in place, for phase 1 lot release, right? And so if we do have a protein, like in this case, there's a protein attached to this PEG, there are some specific requirements, so we need to make sure that we have consistent size and also consistent charge variants on our protein. And so we have 2 specific types of testing. So in this case, because this is not a standard mAb, it's not a standard IgG, I wanted to show you what we needed, what we had to modify with our standard QC testing, because this is also something that the industry is going through at the moment. So in our QC testing labs, we have standard instruments, right? But when we have very unique formats coming down the pipeline, we have to change our system, so we have to adapt the methods. But every time you need to change a method that is already validated, it requires months. It's not like just, you change the method. You have to show that it's comparable to the method that was there before. And so it's something that cannot be done, very fast. So this is like a classical example of what happens for QC testing. So this molecule, so the target is our 8 Fabs per PEG, but because of the inherent polydispersity of the PEG, we have some lower and higher molecular weight variants. And so because we have these variants, so the profile, can, when we do a standard quantitative of the size profile by CSDS, we see all the different variants. And then if we see all the different variants and we know that the size has an impact on the half-life, it is key to show that we have consistent size because we want to have consistent half-life. So the question here was, can we use like a standard QC assay such as CSDS to ensure that we have consistent size? And so that's what we did. So we isolated the different variants, 6, 7, 8, 9, and higher. And then for each one of them, we wanted to measure the size because, as I mentioned before, we have a very good correlation between size and half-life, as we can see here. And so we know that the expected size for our molecule to have the expected half-life is about 10 nanometers. So we need to make sure, can we make sure that with the CSDS we can consistently measure these 10 nanometers? So that's what we did. So here we have all our different variants, and this is the size. So we see, for example, we have about 0.4 nanometer differences per fab conjugated to our PEG. And so we have about, for these variants that we have isolated, there's some differences between 10.5 and 9.7. But also what we know based on rabbit data is that we need to have a difference higher than 1.5 nanometers to have a significant change in half-life. So the next step, so we performed our conjugation, our pegylation at different scales. So we have our research material, then we have our reference standard. We did 1 gram, 7 gram, 20 gram, 300 grams, 600 gram pegylation, really to show that we can scale up. And then we wanted to see what were the ranges of all these different variants. So these are the ranges of the low, of the standard target, and on the high fabs per PEG. And then for them, we have the measured size in nanometers. And so we can say, so these are our expected variability ranges, and we know that the size in those ranges is not, the difference is not higher than that 1.5 nanometers. So we know that this assay then can be used in our expected variability of our manufacturability ranges to measure the size. And so that's what we were going to use to control the size. So the CSDS was going to be used as a surrogate for size. Something else that we did, something that's important also with the proteins, is to ensure that we have consistent profile with the charge variant, so our acidics and our basics. So if we take a protein and we do ICIF, so capillary isoelectrofocusing, or ion exchange chromatography, we can measure the size. But in this case, it's a little bit more complicated because we have 8 proteins attached to the same core. So every time that we want to measure the size, we have an average of all the proteins that are attached to the core. So we can use these to have a nice, like an overall average idea of the charge, but we cannot really use these to give like the right measurement of the charge. So here, just to give you an example, so in green is the protein by itself, and in orange is when it's attached to the core. So it's really clear from this data that we need to use like a different strategy to be able to ensure that we have consistent charge variants. And so what we did is say, so let's see if we cannot use the charge control on our final conjugate, can we show that the process is not changing the charge profile? And because it's lyophilized, we also know that it's stable much more than a liquid formulation. And so that's what we did. So we used the standard charge assay on the protein throughout the process. So we did like a mock conjugation. So our protein went through the process without the PEG. And then we measured the charge on our protein, and we compared the charge profiles at every step. And we also did this upon stability. I'm not showing the data here, but we have observed that there is no significant change in the charge across the process and upon stability. And so we use this approach to say then we can measure our charge profile on the intermediate protein from the beginning, because we have demonstrated that the process is not impacting and also is stable upon time. And so this is also, so the assays, the standard assays, but the thought process is different compared to a standard protein. All right, so this takes me to the conclusion. So I hope in these 15, 20 minutes, I showed you some of the thought process and key considerations that we go through in development when we get the ideas from research, right? So it is key really to start working as early as possible with the research body on representative material, right? So when I mean representative, we need to make sure that we have the raw materials that are of comparable quality of what we're going to use, and also the formulation is important, and the concentration, right? So this is key. Also, manufacturability and concentration range, they really play a key role on the timeline and on the development considerations. And novel technologies or novel control strategies, as I show you for charge and size, are really needed when we use standard QC assay, but with these unique formats. And yeah, these are all the people that I like to acknowledge. So there's strong collaboration. So I come from PAC, protein analytical chemistry, but there's a strong collaboration with drug delivery and also small molecule. And this was the team that was working on this molecule with me.
You mentioned this drug was no longer in the pipeline. So can we ask why after all the hard work? It's a secret question.
I think it's a very competitive space, right? And so probably there's like other companies out there that are doing things that are more advanced. Yeah, that's all I can say. So, but we're working on something else now, yeah, in the same area, but not the same format. But this works well. If you want to increase the half-life, this format works well. Yeah. And we have also published a few papers on these if you want to look them up.
So you mentioned about multiple factors, charge, size, which may impact the retention of the drug in the eye. So what is the most critical factor?
So here is size.
So what is the optimal size for the intravitreal injection?
So to stay in the eye or for the injection?
Yeah, for stay in the eye.
So,
so
this
long
format,
retention.
so 10 nanometers was working well for about 3 months frequency, every 3 months injection. So if you want to target 6 months, probably you have to go a little bit bigger than 10 nanometers. Yeah, so definitely size, it's important. And then charge, it's important for other things, right? So if you do have post-translational modification next to your CDR, so it's the part that it's important to bind to the target, those modifications can impact the way your protein binds to the target, and so might not work anymore, right? So that's why it's really important to make sure that your protein is stable, and then especially in the CDR region, you don't have modification that might impact your mechanism of action, yeah.